Hello viewers, today in this video we are going to discuss ion exchange chromatography. Before diving into the topic, let us first understand what is chromatography. The word chromatography is derived from two Greek words, that is chroma, which means color, and graphene, which means to write. The term was first coined by Mikhail Stwit, a Russian botanist, in 1906. He used chromatography to separate plant pigments like chlorophylls, carotenoids, and xanthophylls, which appeared as a colorful bands on a column. This technique was named based on the visual separation of colors. Chromatography is a separation technique used to isolate and analyze components of a mixture based on their differences in physical or chemical properties such as polarity, charge, or size. It is an important biophysical technique that enables the separation, identification, and purification of the components of a mixture for qualitative and quantitative analysis. Chromatography is based on the principle where molecules in mixture applied onto the surface or into the solid and fluid stationary phase is separating from each other while moving with the aid of a mobile phase. Here, the stationary phase is a fixed medium where the separation occurs. Example, silicogel, cellulose, or liquid coated on a solid support. Mobile phase is the moving phase that carries the sample mixture through the stationary phase. It may be a liquid or gas. Example, water, ethanol, methanol, or acetonitrile are commonly used in paper and liquid chromatography. Helium and nitrogen can be used in gas chromatography. Next, we'll see the basic terms which are used in chromatographic techniques. Elution is the process of moving a compound through the stationary phase using the mobile phase. Elution time is the duration it takes for a specific component of a sample to travel through the chromatography system and reach the detector after being introduced into the column. It reflects how long the component interacts with the stationary phase relative to the mobile phase. Adsorption is the process where molecules stick to the surface of the stationary phase, and it is common in solid phase chromatography. Analyte is the substance or a component in the mixture being analyzed. Retention time is a compound takes to travel from the start of the stationary phase to the detector. Longer retention time indicates the stronger interaction with the stationary phase. Retention factor. In thin layer chromatography, it is the ratio of the distance of a compound travels to the distance traveled by the solvent. Resolution is the ability to distinguish and separate two closely eluting compounds in a mixture. Higher resolution indicates better separation. Gradient elution is a technique where the composition of the mobile phase is gradually changed to improve separation. It is common in HPLC, that is high performance liquid chromatography. Chromatogram, a visual representation of the separation process showing peak corresponding to different compounds. Now let's see ion exchange chromatography. Ion exchange chromatography is a powerful technique used to separate charged biomolecules such as proteins, peptides, and nucleic acids based on their different charges. Ion exchange chromatography works on the principle based on the electrostatic interaction between charged molecules in the sample and oppositely charged groups on the stationary phase. It is basically on charged base separation. Molecule in the sample carries charges depending on their chemical structure and the pH of the mobile phase. These charged molecules interact with the oppositely charged groups on the stationary phase. Based on the charges, we have cation exchange chromatography and anion exchange chromatography. Cation exchange chromatography uses a negatively charged stationary phase, binds positively charged molecules, example proteins with a positive net charge at the operating pH. Anion exchange chromatography uses a positively charged stationary phase, binds negatively charged molecules, example DNA. Next, move on to components of ion exchange chromatography. First one is ion exchange resin is composed of a solid matrix with charged functional groups, example cellulose, agarose, or synthetic polymers. Cation exchanges are negatively charged groups that binds positively charged ions, example sulfonic acid. Anion exchanges are positively charged groups that binds negatively charged ions. Example quaternary ammonium. Buffer solution, it acts as a mobile phase. It is used to maintain a constant pH and ionic strength during separation. Buffers also helps in eluting the bound molecules by altering the ionic strength or pH. Sample is the mixture of molecules to be separated, typically dissolved in a compatible buffer. Chromatography column contains the packed resin through which the sample and buffer flows. It can be either glass, plastic or metal depending on the application. Eluting mechanism is basically of two types, as either isocratic medium or gradient medium. Isocratic medium refers to the technique where the composition of the mobile phase, that is ionic strength or pH, remains constant throughout the process. And the gradient medium is achieved by either salt gradient or pH gradient. Salt gradient is increasing the ionic strength, displaces bound molecules. In pH gradient, changing the pH alters the charge of molecules and their binding affinity. Detection system, it monitors the elution of molecules typically using UV visible spectrophotometry or conductivity detectors. Fraction collector, it collects the separated components as they elute from the column in discrete fractions. Now we'll see the procedure. It starts with equilibration. The stationary phase is equilibrated with a buffer of appropriate pH and ionic strength to prepare for sample binding. Then sample application or sample loading. The sample is loaded and target molecules with an opposite charge to the stationary phase bind strongly. Then the unbound or weakly bound molecules are washed out using the starting buffer. After washing, bound molecules are selectively eluted by increasing the ionic strength or changing the pH. Then the eluted fractions are collected and analyzed typically by UV absorption or other detection methods. Now, then applications of ion exchange chromatography or protein purification, that is isolation of enzymes, antibodies and other proteins based on their isoelectric points, and separation of RNA, DNA and oligonucleotides, and it is also used for water softening process, which is removal of metal ions from water. And in biopharmaceutical industries, it is used for purification of therapeutic proteins and monoclonal antibodies. Advantages of ion exchange chromatography are high specificity and resolution, scalable for industrial applications. It can separate similar molecules based on slight charge differences. And disadvantages are sensitive to pH and ionic strength changes. It requires optimization for each type of sample, and it is limited to charged molecules. Thank you for listening the video.